Audio Jungle.
Importance of homeostasis. A. To maintain a constant internal environment even when the external environment changes. B. Life of the organism becomes less dependent on the external environment because the organism is able to control the changes in its internal environment to compensate for the changes in the external condition. C. Homeostasis ensures an optimum internal environment for the cells to function, the organism to survive and reproduce efficiently. D. Homeostasis enable biological system to function efficiently and smoothly with minimum wastage of energy. E. Organism can live in a wider range of habitats because the organism can live in areas with variable conditions. F. Organism can increase or decrease its metabolic rate to suit its requirement. Thermoregulation when body temperature increases. Thermoreceptors detect the changes, impulse is sent to the temperature regulatory center in hypothalamus for physiological responses and cerebral cortex for behavioral responses. Then, impulse is sent to the effector to bring about changes back to normal. Physiological control when body temperature increases. Number 1, sweat glands increase sweating. Number 2, hair erector muscle relax and flatten hair. Number 3, vasodilation of skin arterioles. Number 4, no shivering of skeletal muscle. And number 5, metabolic rate decreases. Thermoregulation when body temperature decreases. Thermoreceptor detect the changes. Impulse is sent to the temperature regulatory center in hypothalamus for physiological responses and cerebral cortex for behavioral responses. Then, impulse is sent to the effectors to bring about the changes back to normal. Physiological control when body temperature decreases. Number 1, hair erector muscle contract and raises hair. Number 2, vasoconstriction of skin arteriole. Number 3, shivering of skeletal muscles. And number 4, metabolic rate increases. carbohydrate metabolism. In glycolysis, glucose is oxidized to pyruvate before entering the Krebs cycles. Carbohydrate metabolism. In glycogenesis, A. When the blood glucose level rises above its normal level, the beta cells of the islets of Langerhans are stimulated to secrete hormone insulin. B. Insulin converts excess glucose to glycogen. 1. Insulin attached to insulin receptor of the hepatocyte. 2. Glucose is transported into the hepatocyte via glucose transporter 4. Number 3. Glucose is phosphorylated to glucose 6-phosphate. Number 4. Glucose 6-phosphate is isomerized to glucose 1-phosphate. Number 5. Glucose 1-phosphate is dephosphorylated to UDP glucose. And number 6. Glucose is polymerized to glycogen as glucose is removed from UDP glucose. C. Glucose is stored in the liver and muscles. Fructose and galactose are converted to glycogen and stored in the liver. D. Insulin increases the rate at which excess glucose is converted to fats and then stored in adipose tissue, under the skin and around internal organs. E. Insulin inhibits glycogenolysis and gluconeogenesis. Carbohydrate metabolism. In glycogenolysis, A. When the blood glucose levels falls below its normal level, the alpha cells of the islets of Langerhans are stimulated to secrete hormone glucagon. B. Glucagon activates glycogen phosphorylase, which increases the rate of glycogenolysis, the conversion of glycogen to glucose. C. In time of stress, hormone adrenaline or epinephrine is released by adrenal gland, and noradrenaline or norepinephrine is released by endings of sympathetic neurons. This stimulates glycogenolysis to increase supply of glucose to the cell. In carbohydrate metabolism, gluconeogenesis. A. Glycogen reserves are limited in body cells, and when during strenuous exercise or fasting, there is an increase in the synthesis and release of glucocorticoid hormones. B. Glucocorticoid hormones stimulate gluconeogenesis, the conversion of lactate, pyruvate, glycerol, and glycogenic amino acids to glucose. C. There are three pathways in gluconeogenesis. The first source is from lactic acid or Cori cycle. The second source is protein, amino acids. And the third source is triglycerides. Cori cycles refer to the metabolic pathway in which lactate produced by anaerobic respiration in the skeletal muscle is transported by bloodstream to the liver to be converted into glucose. In the liver, lactate is oxidized to pyruvate. The pyruvate is reduced to glucose 6-phosphate and then dephosphorylated into glucose. The glucose produced may be stored in the liver as glycogen or may be transported in the blood to the skeletal muscle cells for cellular respiration. Cori cycle prevents accumulation of large amount of lactic acid, which causes lactic acidosis and a fall in blood pH below its normal level. For protein amino acids, there are three ways. Number 1, glycine, alanine, serine, and cysteine are converted to pyruvate directly. Number 2, lysine is converted to acetyl-CoA indirectly. And the third way is some amino acids form intermediates in Krebs cycle. Aspartic acid is converted to alpha-ketoglutarate, while glutamic acid is converted to oxaloacetate. Triglycerides is hydrolyzed to glycerol and fatty acids. Glycerol is converted to pyruvate, while fatty acids undergo beta oxidation to form acetyl-CoA. In protein metabolism, transamination is used to synthesize non-essential amino acids. Number 1, it refers to the transfer of amine group from one molecule to another. Transfer of amino group between amino acids and keto acids. Number 2, alpha-ketoglutarate and alanine are transaminated to glutamate and pyruvate. In protein metabolism, deamination of excess amino acids into keto acids and ammonia. Number 1, deamination is the oxidative reactions that occur under aerobic condition in all tissues, but especially the liver. Number 2, the body cannot store excess proteins or amino acids. Number 3, the excess amino acids are taken up by the hepatocytes and deaminated. Number 4, deamination involves the removal of the amino group from the amino acid to form ammonia. Glutamate is deaminated to alpha-ketoglutarate and ammonia. urea formation, ornithine cycle. Ammonia produced is then converted into urea through ornithine cycle. Ammonia produced by the deamination is soluble and toxic. Therefore, it must be converted to urea in the liver and then transported to the kidney where it is excreted. The conversion of ammonia into urea occurs in a cyclic reaction known as ornithine cycle. Number 1. The first step in the production of urea is ammonia from deamination enters the mitochondrial matrix of liver cells, react with carbon dioxide and water to form carbamoyl phosphate. 2-ATP is used in this process. Number 2. Carbamoyl phosphate then reacts with ornithine to form citrulline in the mitochondrial matrix. Number 3. Citrulline then diffuses into the cytoplasm of liver cells and react with aspartate to form arginosuccinate. This reaction uses an ATP. Number 4. Arginosuccinate cleaves to form fumarate and arginine. Number 5. Arginine is hydrolyzed, releasing urea and producing ornithine, which can begin the cycle again. Number 6. Urea is then carried in the bloodstream to kidney to be excreted in the urine. Ultrafiltration. Blood containing nitrogenous waste enter kidney via renal artery. Diameter of afferent arteriole is larger than that of efferent arteriole. This creates a high hydrostatic pressure in the glomerulus. The high hydrostatic pressure in glomerulus forces blood content to filter into Bowman capsule through ultrafiltration. Glomerular filtrate contains glucose, amino acids, vitamins, hormones, urea, uric acid, creatinine, ions, and water, except plasma proteins, blood cells, and platelets, which are too large to pass through the wall of glomerulus. Selective reabsorption in proximal convoluted tubule. From Bowman capsules, glomerular filtrate flows into proximal convoluted tubules where active reabsorption of useful substances occurs. All glucose, amino acids, and a large proportion of mineral salts are actively reabsorbed into the peritubular capillaries. A large proportion of water, over 80%, is reabsorbed by osmosis. Some urea is reabsorbed from filtrate by diffusion. Waste-soluble substances, example creatinine, and drugs which are not filtered in glomerulus are carried by blood to proximal convoluted tubule and actively secreted into its lumen. Selective reabsorption in loop of Henle. The descending limb of loop of Henle is permeable to water. The ascending limb of the loop of Henle is impermeable to water. Sodium ion and chloride ion are actively transported out of the thick segment of ascending limb into the medulla tissues. This lowers the water potential in the medulla. Water diffuses out from the descending limb of loop of Henle through osmosis. As filtrate flows out to the thin segment of the ascending limb, sodium ion and chloride ions are passively diffused out of the thin segment of ascending limb into the medulla tissues. More water diffuses out from the descending limb of loop of Henle. This is the countercurrent multiplier mechanism which takes place. selective reabsorption in distal convoluted tubule. Remaining salt or water is reabsorbed in distal convoluted tubule and collecting duct into its surrounding blood capillaries by hormone regulation and depends on the body needs. Active secretion. Urea, toxic substances, and drugs are actively secreted into distal convoluted tubule from surrounding capillaries. Filtrate flows into collecting duct as urine and it contains water and excess mineral salts not required by body and nitrogenous-based substances such as urea, ammonium ions, uric acid, and creatinine. Antidiuretic hormones, ADH. Number 1, when the blood osmolarity or osmotic pressure is higher, the change is detected by osmoreceptor in the hypothalamus. Number 2, nerve impulses are generated and sent to the posterior pituitary gland to stimulate the release of antidiuretic hormone, ADH. Number 3, ADH is produced by the hypothalamus and transported to the posterior pituitary gland to be stored. Number 4, ADH is released by the posterior pituitary gland to its target organ, the kidney. Number 5, ADH binds to its receptor in the plasma membranes of epithelial cells lining the distal convoluted tubule and collecting duct. Number 6, it activates a series of enzyme control action. Enzyme phosphorylase becomes activated. Number 7, ADH increases the permeability of the wall of distal convoluted tubule and collecting ducts to water. Number 8, the phosphorylase causes vesicles with aquaporins, protein water channels, to move to the epithelial cells. Number 9, the vesicle fills with the plasma membrane. Number 10, water then moves freely through aquaporins down its water potential gradient from the collecting ducts into the surrounded concentrated medulla interstitial fluid and then into the blood capillaries. aldosterone. Number 1. Activation of renin-angiotensin-aldosterone system, RAAS. Number 2. When blood pressure or volume drops, the juxtaglomerular apparatus or complex releases the enzyme renin. Number 3. Liver releases angiotensinogen into the blood. Number 4. Renin catalyzes the production of the polypeptide angiotensin 1 from the protein angiotensinogen. Number 5. Angiotensin converting enzyme ACE converts angiotensin 1 into angiotensin 2. Number 6. As a hormone, angiotensin 2 triggers vasoconstriction, increasing blood pressure. Number 7. Angiotensin 2 also stimulates the adrenal gland to release a hormone, aldosterone. Aldosterone stimulates sodium-potassium pump in cells of distal tubules and collecting duct to reabsorb more sodium ions and water from tubules into blood. And number 9, thus increasing blood volume and blood pressure increases back to normal. Importance of transpiration. Produce a force called transpiration. Pull on the xylem of the stems and roots, uplift water to the leaves. Transport of mineral ions dissolve in water. Transport of water-soluble organic products such as the plant hormones, especially ethylene and cytokinin. Helps to cool the leaves through evaporation. Stimulates the development of mechanical tissues in plants. Cell walls become more compact, thicker, and cutinized. Maintains an optimum degree of turgors in cells. Improve the taste of fruits. Solutes inside the cell became more concentrated. Concentration of sugar solution in the cells of fruit increases the fruit taste sweeter. Halophytes. Its morphological and anatomical adaptations are: A. The presence of hydatodes at the edge of leaves to remove excess salt. B. The new metaphors are modified roots for gaseous exchange. C. The plant has prop root or buttress root to support and hold plants in muddy soil. D. The leaves are small or large with thick layer of waxy cuticle to prevent evaporation. And E. The leaves have sunken stomata, reduced in number, and fine hairs on leaf surfaces. Halophytes. The physiological adaptations are: A. Root cells accumulate mineral salts by active reabsorption of salt to avoid cell plasmolysis. B. The plants produce more abscisic acids to close stomatopores to avoid water loss by transpiration. C. Mangrove plants have vivipary germination to increase the survival of species. (05) Morphological and anatomical adaptation. Number 1. Have smaller or needle-like leaves to reduce the total surface area. Number 2. Have sunken stomata to reduce evaporation. Number 3. Have succulent stem or leaf to store water. Number 4. Have very shallow and extensive root system. Superficial root to exploit water condition at night on soil surface. Number 5. Have thick waxy cuticle covering leaf and stem to reduce water evaporation. And number 6. The stomata closed during daytime or stomata open at night for carbon dioxide uptake to reduce transpiration. 05. Physiological adaptation. Desert plant practice CAM pathway in dark reaction of photosynthesis to ensure enough supply of carbon dioxide via stomata pore, but still able to prevent excess water loss through the same opening. This type of plants open stomata pore during the night to absorb carbon dioxide from the atmosphere. Night is cold in desert area, no evaporation of water, therefore no water vapor escape through the stomata opening. Thus, this will prevent water loss. During the day, stomata pore closed, but the presence of sunlight allows the process of photosynthesis to occur.